Okay, so we talked about predicting subcellular localization with an SVM. Thank you. So there's only you who... Ole? Where are you from?
Sweden.
So sorry for getting it wrong. So only Ole, we make a photo afterwards. So let's get back to this. We built a tree with the nodes being SVMs, and the nodes make a decision, a binary decision, is it that or that class? And we begin with the top of the node that is a relatively easy to do decision, and it is in fact sort of mirroring the biological tree as I showed here. Now, so the first question here is essentially, am I secreted or not? And according to yes, if yes, we go and pass in this direction, and then for every single step, there's an SVM that makes a decision. Okay, so this is one way of doing it. What I skipped some slides. I skipped a lot of slides. No? Now, the disadvantage we said of this is a decision I make up here, I cannot, any mistake I make in the high order, I cannot recover. So I will, if I go wrongly to the left, I will end up making a decision somewhere. And that will be a wrong decision simply because the first decision was wrong. There's no way to recover from that. The beauty of this is, well, one, for the formalism in SVM, this is a way to realize a multi-class classification. The other beauty of it is that at every single step here, you can focus on different input information. I told you that proteins that are secreted have a signal peptide, or many proteins that are secreted have a signal peptide, and that the signal peptide is at the beginning of the protein, the first roughly 30 to 50 residues. So this SVM can just look and focus on that region, or at least one of its input could be a zoom into that region, in fact the composition only for that region, and the rest could be the amino acid composition for the rest of the protein. While some others can look at other features that are relevant. For instance, in the nucleus, we know we have prevalence of positive charges. And again, so we could have a special one that is a unit that is simply looking at the positive charge, the sum of the positive charges. There are other ways of doing it. Mitochondria, again, there is at the beginning of the protein, there's a terminal signal. Now, briefly, there are many other methods out there that do it, and that typically is the situation when you have a data set and you can apply machine learning. So once somebody has done it, many people will try to copy that with different machine learning tools very often, sometimes with different ideas in terms of how to use the input. So the initial tools really only used amino acid composition, so essentially had 20 input features, right? Then came these methods such as p-sort, and there's the latest version, Wolf p-sort, that essentially combines motifs, targeting motifs. So at the beginning there's a signal peptide. If I find that, I take that. Nuclear localization signals, I have a database of nuclear localization signals. If I find that, I take it. And then it applies amino acid composition to whatever remains. Okay? The signal P, target P, we already talked about. Is N-terminal sequences, or N-terminal, the signal that is really in the beginning of the protein. Most methods somehow use a derivative of sequence composition to something that is an overall feature. And I believe, I'm not going to go into that, I'm going to skip this. When I skip things, then they will also not ask in the exam. So here is another kind of play on this motif, Sherloc. So the first part is I talked about this method lock-key that essentially takes the keywords about function, grinds them up, and tries to predict subcellular localization from those keywords. And this is part of what it does, but it does this using a PubMed abstract. Simply a paper about a protein, and in that paper it tries to read whether the subcellular localization is in this abstract, grinds up the entire abstract and sees what are sort of the keywords in a published paper that would reveal the subcellular localization. And then it combines this with a bunch of de novo methods. Some of these methods here use target. This implies, again, the N-terminal signal, so the beginning of a protein, there's a signal. Some of them here, they in fact try to look at trees and for evolutionary relation and see whether one of those in this tree has an experimentally annotated structure. This is ultimately homology-based inference. I talked about that. And some of them try to find motifs. Motifs are the kind of thing like NLS. So other motifs. And then there are different SVMs that use different amino acid composition. Here that uses secondary structure and solvent accessibility. So it uses different features, and all of these are different methods. And then they are all combined with in a profile vector, and then it essentially does something similar to the tree that I showed you, a one against many. Yes?
Just a quick question. Why does it just read the abstracts and not the entire paper?
It says full abstract. Why does it do?
Yeah.
So there are two reasons for that. First, the sense is, so text mining, mining human readable text is not easy. Okay, statement number one. Statement number two, if you try to make it as easy as possible, where in the paper do you believe you have the information that is most condensed or most... the idea really is that in the abstract, the human makes an effort, who writes this paper, makes an effort to put everything important into the abstract. So the hope is that by just understanding 500 words in the abstract, or less than 500 words, 200 to 500 words, we sort of get the main theme of the paper without sort of having to distinguish what is methods, what is, and all of these details. That's one reality. The other reality, however, is even more important. Abstracts are available for every single paper through PubMed. For every single paper in molecular biology. Full papers are not. So currently, in fact, it's still true. If you have a special arrangement here with the university library, you can access full papers, but it is actually not easy. So we have some deal, but to maintain that deal is not easy. And ultimately, that means that despite our ability to use the university library, ultimately at the end of the day, when we do full text in our group, we actually use open access journals. So we begin to focus really in our text search on open access journals, which in fact is an interesting turn, because it completely, for people who do text mining, the open access journals have completely begun to dominate all of these text mining. But these are the two main reasons for using abstract, and ultimately the first one really was availability. And then, by the way, there's another issue here, going through the entire PubMed, just the abstracts. If you do a hash for a keyword, so if all you actually want to do is find the word nuclear, then that's trivial. But you're trying to understand sentences, and at that point it gets actually also CPU intensive. So running the entire PubMed, so for some of the methods takes in the order of months. Just the abstracts, not the papers, right? So there are a couple of reasons. Okay, so this is the idea here. It comes from a group, from Harald Shakai and Oliver Kohlbacher. It's a collaboration between the US and Tübingen. Underneath is an idea of doing a rather interesting or a new way of using SVMs by simply using a lot of different features. So stacking up SVMs by pre-digesting the information. In some sense, so you use individual features in a different way and put them all as input into the SVM. In some sense, this is a way of an expert trying to do something such as deep learning or for an SVM. Because there are different modules that pick up different signals, and these signals are then passed on to a higher level module. This is essentially exactly what you do in deep learning. Okay, here in CELLO, the trick really is in the coding. So it's a partition into k segments of length L, and in these k segments, you use the composition for each. And then you hope that by the aggregation of all of these segments, you get a signal. And in fact, it allows motifs with gaps. And looks at a sliding window composition. So the tool in itself is not that successful, but the idea really is very different. Okay.